Welcome. In this clinical review, we're going to break down the core electrophysiological principles of nerve and muscle function. This is really the essential foundation you'll need for understanding therapeutic electrical stimulation in your physiotherapy practice.
Okay, so we're going to trace the entire signal pathway. We'll start with the cell's resting state, then see how an action potential is generated and how it's transmitted. From there, we'll look at the functional motor unit and how we control force. And finally, we'll wrap up by analyzing the critical clinical consequences when that signal is lost during denervation.
All right, let's get started. First up, the resting state. We really need to understand this concept because it's what sets the stage for everything that follows. It creates the specific electrochemical environment that makes a cell excitable in the first place.
So what exactly is the resting membrane potential, or RMP? You can think of it as a state of electrical polarization across the cell membrane. In a typical skeletal muscle cell, this is about negative 90 millivolts. The key takeaway here is that the inside of the cell is negative relative to the outside. This negative charge is absolutely essential for the cell to be excitable. It's the stored potential energy that's waiting to be unleashed for depolarization.
Okay, so how is this resting potential established? Well, there are two main players at work. First, and this is the major contributor, is potassium efflux. The cell membrane is highly permeable, or leaky, to potassium ions. Since there's a high concentration of potassium inside the cell, it naturally diffuses out, taking its positive charge with it. This leaves behind unpaired negative anions, making the cell's interior negative. The second key mechanism is the sodium-potassium pump. Now, this is an active transport system. It's constantly working, pumping 3 positive sodium ions out for every 2 positive potassium ions it brings in. This results in a net loss of positive charge, but its most critical role is maintaining the ion concentration gradients that the entire system relies on.
All right, so we've established that the cell is primed and ready at its resting state. Now let's look at how it uses that potential to fire off an electrical signal, the action potential.
This whole process follows what's known as the all or none principle. Once a stimulus pushes the membrane to its threshold, that's around negative 55 millivolts, a stereotyped cascade kicks off. First, you get depolarization. Voltage-gated sodium channels fly open, and sodium ions rush into the cell, making the inside positive, shooting all the way up to about plus 30 millivolts. Next comes repolarization. Those sodium channels inactivate, and now slower voltage-gated potassium channels open. Potassium flows out of the cell, restoring the negative potential. Finally, there's often a brief hyperpolarization, a little overshoot, where the membrane becomes even more negative than resting potential, simply because those potassium channels are a bit slow to close.
And this graph here shows you that entire event mapped over time. You can see it starts at the resting potential, a stimulus hits, and we climb to the threshold. Then bam! That sharp spike is the depolarization from the sodium influx. The downward curve is repolarization as potassium leaves the cell. You can even see that little dip of hyperpolarization at the bottom before it levels back out. The incredible thing is that this whole complex event happens in just a couple of milliseconds.
Now, this brings us to a really important concept, the absolute refractory period. Right after an action potential fires, there's a brief window where the voltage-gated sodium channels are in an inactivated state. They're not just closed, they're inactivated and cannot be reopened no matter how strong the stimulus is. Why is this so important? Well, it acts as a crucial safety mechanism. It ensures the signal can only travel in one direction, forward, and it also sets the upper limit on how fast the neuron can fire. Okay, so we've successfully generated a signal, but how does that signal travel down a long nerve fiber? And then how does it get passed on to a muscle? That's what we'll cover next, propagation and the synapse.
When we talk about how an action potential travels, there are really two main modes. In unmyelinated axons, you have what's called continuous conduction. The action potential has to be regenerated at every single point along the membrane. As you can imagine, this is a relatively slow and metabolically expensive process. But in myelinated axons, we have a much more efficient system, saltatory conduction. The myelin sheath acts as an electrical insulator, so the signal effectively jumps from one gap, called a node of Ranvier, to the next. This is significantly faster and much more energy efficient.
At the very end of the motor neuron, we find the neuromuscular junction. This is a highly specialized chemical synapse, and its entire job is to convert that electrical signal, the action potential, into a chemical signal to pass the message to the muscle fiber. You can see the key structures here. The presynaptic axon terminal, filled with vesicles of acetylcholine, the tiny gap called the synaptic cleft, and the postsynaptic motor end plate on the muscle fiber, which is packed with nicotinic acetylcholine receptors.
So let's walk through this sequence of events. First, the action potential arrives at the axon terminal. This arrival triggers voltage-gated calcium channels to open, and calcium floods into the terminal. This calcium influx is the critical trigger that causes the synaptic vesicles to fuse with the membrane and release their acetylcholine. The ACH then diffuses across the cleft and binds to its receptors on the muscle fiber. This opens ion channels, creating a localized depolarization called an end-plate potential, or EPP. If that EPP is strong enough to reach threshold, it triggers a full-blown action potential in the muscle itself. In a healthy system, the EPP is actually much stronger than needed. This is called the safety factor and ensures a reliable transmission every single time.
Okay, the signal has now successfully jumped to the muscle fiber. But the nervous system doesn't control muscle force by activating individual fibers one by one. Instead, it works with functional groups called motor units.
So what exactly is a motor unit? It's defined as a single alpha motor neuron in all of the muscle fibers it innervates. Think of it as a single functional team. When that one neuron fires an action potential, every single muscle fiber on its team contracts together in perfect synchrony. This is the smallest increment of force that the central nervous system can command.
Now, not all motor units are created equal. Their size, which we can define by the innervation ratio, or the number of fibers per neuron, is directly related to the muscle-specific job. For muscles that require very precise, fine motor control, like the extraocular muscles, you'll find very small motor units with just a few fibers per neuron. In contrast, for large postural muscles that generate powerful contractions like the soleus, you'll see huge motor units with hundreds or even thousands of fibers per neuron.
So, this raises a key question. If motor units fire in an all-or-none fashion, how do we produce smooth, controlled movements? How can we go from gently picking up a cup to powerfully lifting a heavy weight? The answer lies in how the nervous system grades contraction to control force.
The key to this is a fundamental concept in motor control known as the size principle of recruitment. This principle states that motor units are always recruited in a very specific, orderly fashion, from the smallest to the largest. What's fascinating is that this isn't some complex command from the brain. It's a result of the intrinsic properties of the motor neurons themselves. Smaller motor neurons are simply more excitable. They have a higher input resistance, so a given synaptic input will bring them to threshold first. It's an incredibly efficient and automatic way to grade muscle force.
This table really summarizes the correlated properties nicely. The smallest motor units, which get recruited first, consist of type 1 or slow-twitch muscle fibers. They're characterized by low force generation but are highly resistant to fatigue, which makes them ideal for postural control and endurance tasks. As force requirements increase, the larger motor units are recruited. These are comprised of fast-twitch type 2 fibers. They generate progressively greater force, but they're also much more fatigable.
For our final section, we're going to shift to a critical clinical topic. What happens when that connection, that signal from the nerve to the muscle, is lost? We're going to examine the pathophysiology of denervation.
When a muscle is denervated, a predictable and devastating cascade of events begins. The initial loss of neural input stops not only muscle activity, but also the delivery of essential neurotrophic factors. Within weeks to months, the muscle fibers begin to atrophy, decreasing in size and strength. If this process continues long-term without reinnervation, the muscle fibers are progressively destroyed and replaced by non-contractile fibrous and adipose tissue. Eventually, this can become irreversible and lead to contractures. This comparison is fundamental for applying therapeutic electrical stimulation. With a healthy, innervated muscle, we typically stimulate it indirectly by depolarizing its motor nerve. Since nerves are very excitable, this only requires a low-intensity, short-duration pulse to get a brisk, coordinated contraction. But with the denervated muscle, the nerve is out of the picture. We have to stimulate the muscle fiber membrane, the sarcolemma, directly. This membrane is much less excitable, so it requires a high-intensity, long-duration pulse to elicit a contraction. The resulting contraction is also characteristically sluggish and vermicular because the action potential propagates slowly along individual fibers, rather than being activated synchronously as a motor unit. And this brings us to our final, thought-provoking question, which directly links all these physiological principles to clinical decision-making. A thorough understanding of the differences in membrane excitability, the strength-duration characteristics of nerve versus muscle tissue, this is paramount for us as physiotherapists. It's this knowledge that informs our selection of the appropriate electrical stimulation parameters, like pulse duration, intensity, and frequency, to achieve specific therapeutic outcomes,
whether that's neuromuscular re-education of an innervated muscle or attempting to mitigate atrophy in a denervated